Hi, Kelsey here. I'm here to explain my concentration map of this week's definitions. So I'll go ahead and pull that in for you. So everything kind of starts with the plasma membrane, as you can see. From the plasma membrane, the fluid mosaic model was basically made, and in the fluid mosaic model, you can basically see everything that kind of falls under. You can see the phospholipid bilayer. You can see proteoglycans. You can see glycolipids. You can see glycoproteins. You can see those hydrophilic heads and those hydrophobic tails. You can even see certain membrane proteins in that fluid mosaic model. So it's a very common, very popular model, and you can just basically see everything there. So going further down, so phospholipid bilayer, of course, connected to the plasma membrane. It's the main component of the plasma membrane. Again, seen in the fluid mosaic model. Off that phospholipid bilayer, you have glycolipids that are connecting to that phospholipid bilayer. The glycolipids are, of course, connecting to the lipid part of the phospholipid, and they are basically lipids with a carbohydrate group attached. With the glycolipids, you have lipid-linked membrane proteins, so those connect there and then brings you back to membrane proteins, which again brings you back to plasma membrane, so kind of like a big circle there. You also have glycoproteins, which are, again, a protein that has a sugar attached to it, and there's also protein-attached membrane proteins. So the glycoprotein not only is considered a protein-attached membrane protein, but it's also part of that phospholipid bilayer, and again is connected to the membrane protein. So another circle there as well. Membrane proteins have transporter proteins, which they transport materials across the plasma membrane, so back to plasma membrane. You also have anchor proteins, which they anchor membrane macromolecules onto either side of the plasma membrane. So again, back to plasma membrane. Then you have receptor proteins, which detect chemical signals in the environment, and they tell those membrane proteins tell the plasma membrane about what's going on outside the cell. Then you have enzyme proteins, which catalyze covalent bond breakage. It's another membrane protein that goes on inside the plasma membrane. You also have integral proteins, which are proteins that are transmembrane proteins. They're attached to the plasma membrane permanently, so still going back through those membrane proteins back to the plasma membrane. Then you have peripheral proteins, which are temporarily attached to the plasma membrane. So the only difference between these two is this one's permanently, this one's temporary as far as its attachment to the plasma membrane. From there, you have monolayer associated membrane proteins. So the monolayer associated membrane proteins can only be disrupted with detergents. So I know those were kind of far apart, but I didn't realize the connection until after I'd already wrote them that far apart. So I just wanted to make sure it was known that these can only be disrupted by detergents. They are also entirely in the cytosol and exposed to the surface of that plasma membrane on top of it. From there we have, so if we want to go this way, we can go to the detergents, and from the detergents, we know that they are amphipathic molecules, and the detergents actually, sorry, I lost my spot. The detergent molecules have polar heads and hydrophobic tails, so they're almost just like a phospholipid bilayer. So I thought that was pretty crazy as well. But so they have that. They also used to lyse cells. So that's that part. So phospholipid bilayers are amphipathic molecules, which are molecules that basically have a hydrophobic and a hydrophilic part to them. So they have both. So that's why they are considered a phospholipid and a detergent. They're also considered a fatty acid. And there's two types of fatty acids that we talked about in this section, and that was unsaturated and saturated. The difference in unsaturated and saturated is that unsaturated has at least one double bond with the fatty acid chain. So, going on from there, sterols are also an amphipathic molecule, and they are also come off of the phospholipid bilayer in the plasma membrane. Going from there, okay, so there's a big line all the way through, and it talks about ion channels. So ion channels, they are proteins or protein complexes, and they facilitate movement through the membrane. And through these ion channels, there's different kinds. There's membrane channels, there's gated ion channels. But what I thought was pretty neat with the membrane channels is that they go across the electrochemical gradient. So those two kind of work together, which then this also brings you to channel mediated, which they also can use either the concentration or the electrochemical gradient. So I'm going to just jump up to concentration gradients and work back down. So plasma membranes, concentration gradients work across that plasma membrane. So everything that occurs with the occurs along the plasma membrane. With concentration gradients, you have simple diffusion, you have osmosis, which in osmosis, it's the isotonic, hypotonic, and hypertonic solution. So basically cell shrinkage, cell swelling of cells, things like that, all that occurs within that plasma membrane. You also then have passive transport, which with passive transport, I thought that was, it moves along the concentration gradient, but it also can be channel mediated with the passive transport. Passive transport and active transport also help tell you your selective permeability. So I think I've covered where everything connects, but if not, I'll move a little bit closer so you can get a better image of everything. And that completes my flowchart of my definitions for this week. Thanks.